DNA extraction is a useful technique in biology. In today's exercise, we will be comparing the quantity of DNA extracted from 3 different fruits. I have selected strawberries, blueberries, and red peppers. The amount of DNA recovered will be affected by the size and number of chromosomes contained within each kind of plant cell. Humans have 2 copies of each type of chromosome, and there are 23 pairs of chromosomes, giving us a total of 46 chromosomes. Strawberries, on the other hand, are octoploid, meaning they have 8 copies of each chromosome, although there are only 7 chromosomes total for strawberries, giving them a grand total of 56 chromosomes. The number of chromosome pairs in peppers seems to be roughly 12, and typically they are diploid, giving them a total of 24 chromosomes. However, there does seem to be some variation between species and particular cultivars. Similarly, blueberries have multiple varieties, which could be diploid, tetraploid, or even hexaploid, so the total number of chromosomes is difficult to determine. In order to maintain consistency, we will be comparing the amount of DNA recovered from precisely 5 grams of material from each species. Please pause the video now and write down a hypothesis predicting which plant material will contain the most DNA. Between us and the DNA is the cell membrane as well as the nuclear envelope. Recovering the DNA will require first disrupting these lipid membranes. Lysis solution contains dish detergent and salts. The molecules within the dish detergent are amphipathic. Amphipathic molecules allow lipids to mix with water. The salt provides chemical balance by matching the solute concentration typically found inside the cell. For each sample we are testing today, we will add precisely 10 milliliters of Lysis solution. Before we may extract the DNA, we must subject the cells to mechanical disruption. To do this, add your sample to a clear Ziploc sandwich bag. Make sure there is no excess air in the bag. Add your 10 milliliters of Lysis solution, and then begin disrupting the cells with your hands, squeezing them and mashing them until you find a homogeneous slurry. This usually takes 1 to 2 minutes. Once your fruit has been mashed to your satisfaction, we need to strain out the pulp. The lab procedure calls for cheesecloth, which is an excellent strainer for this purpose. However, as I have no cheesecloth on hand, I will instead be using these mesh bags. A kitchen strainer could also be used for this procedure. Once the contents of the bag has been emptied into the tube, record the volume in the results section of your lab report. Here I'm using a ruler to measure the amount of liquid in each tube, and I'm marking it with a grease pencil. I will later have to add an equal amount of ice-cold alcohol to the solution in order to precipitate the DNA, so I'm making a second mark in order to indicate where the fill line will be. To compare the amount of DNA from multiple plant sources, I'll need to repeat the procedure with the other 2 samples. Strawberries and blueberries are relatively easy to mash with just your fingers. However, I have found peppers to be somewhat more stubborn. Therefore, in order to extract the DNA from this pepper, I will require a tool called a mortar and pestle. The mortar is the bowl-like portion of the tool, and the pestle is the rod-like portion of the tool. Using the mortar and pestle, I can grind the pieces of pepper into a much finer slurry than I would have been able to achieve with my fingers alone. Here I'm using the excess Lysis solution to rinse out the mortar. I'm also taking care to ensure that the total amount of Lysis solution used is 10 milliliters at the end, so it will be the same as the other bags. The liquid contained inside the test tube on the far left, with the reddish-orange color, is the red pepper solution. The light brown liquid contained inside the test tube in the very center is the blueberry solution. And the liquid contained inside the test tube on the far right, with a somewhat light pink color, is the strawberry solution. The next step of the procedure is to add an equivalent volume of alcohol to each tube. Either ethyl alcohol or isopropyl alcohol will work well for this purpose, provided that first it has been chilled in the freezer down to an ice-cold temperature. In this video, I am using isopropyl alcohol. As you can see here, the clear alcohol forms a layer on top of the solution inside of each tube. The DNA will precipitate at the interface of these two layers. It takes approximately 5 minutes for the DNA precipitate to begin to form. However, since I am using time-lapse photography, you should be able to see it form in a much more convenient time frame. DNA has a whitish-gray color and a wispy, translucent consistency. If you turn your attention to the test tube on the far right, which contains the strawberry solution, you can already see the DNA starting to poke out. That bubble-like structure that's poking out just above the blue test tube rack, that is the DNA beginning to coalesce. Comparing the 3 test tubes side by side, you can see that each one formed a DNA precipitate. In your lab notebook, mark down which one you think had the most DNA, the second most DNA, and the least DNA. Consider whether this result is consistent with the hypothesis you wrote down towards the beginning of the video. Consider that we are only measuring the DNA using a simple visual comparison. What sources of error do you suppose might come about as a result of this tactic? Consider for a moment whether there is a method that we could use to more accurately measure the amount of DNA contained inside each type of plant tissue. Taking a close look at the DNA in the strawberry tube, we notice that it is actually floated up to the top and is no longer at the middle. Taking an instrument such as a bamboo skewer or a clean pipette, you can actually manipulate the DNA inside of the tube. As I swirl the pipette around inside the tube, you can see that the DNA is actually clinging to it, much like strings of spaghetti on a fork. The DNA is a polymer of nucleotides, so it too is a long string. Next, I thought I would try adding a drop of methylene blue dye to the top of each test tube. Methylene blue dye is exceptionally good at staining nucleic acids such as DNA. When I do this, rather than the dye spreading out to a uniform distribution, you can see that it seems to bind to wispy strands inside of the alcohol. While the bulk of the DNA has formed a single large clump, there are still loose wispy strands of DNA floating around inside the isopropyl alcohol.